Hello class. Today we're going to do an online lecture looking at the primate family tree. What I'd like you to do is take a minute, pause the video, and write down our objectives and keywords. When you've got them all written down, go ahead and hit play. So one other thing that you can do to help prepare yourself for this presentation is print out this primate family tree handout. I've posted the handout as a PDF on our course website, and this divides the primate family tree into the relative groups we'll be talking about today. Once you're able to print this out and start filling in the gaps, you can include the extra descriptions that we use, and you'll see we're particularly interested in understanding how to differentiate between each of these groups. So if you haven't yet, go ahead, pause the video, take a minute to print out this sheet, and then fill it in as we go through the lecture. So what are the kinds of primates? Well, the human primate is just one of over 200 different kinds or species of primates on Earth today. This image comes from your book, and it shows this diversity of species that we have based off of the Linnaean taxonomy of the primate order. You've got it divided from each stage, from the order down to the suborder, the infraorder, etc. And down below, you have images of some examples of the different genuses that are out there. Now, if you look, you'll see that several of these have green stars. I've gone through and tried to identify any of the primate genuses and species that we have represented at the St. Louis Zoo, since I'm going to have you guys do primate observation at the zoo. So if you have a chance to come back to this slide, I'd recommend taking a look at it before going to the zoo, just to get an idea of who we have represented. Now I'll tell you, we have great representation among lemurs. So when we talk about lemurs today, think about whether or not you're interested in doing observation with them. They're also in an indoor habitat, so if it's raining, you might want to go hang out there. And then we also have a ton of representation among the greater apes, which are on the far right side of this image. So if you're interested in some of the primates that are most closely related to us, that's an opportunity for you to check those out. Today we're really going to focus more on what's happening before we get to the apes. And so we'll be looking at the monkeys, and then the lemurs, lorises, and tarsiers. Now this shows our position using the basic Linnaean taxonomy. As you saw from our previous slide, the basic Linnaean taxonomy has gotten a lot more complex. So in addition to these steps of species, genus, family, order, class, phylum, and kingdom, we've added many steps in between. So we have a subspecies, we're a suborder, we have a paraorder, etc. Our position using the basic Linnaean taxonomy is that we are animals with a backbone, so chordates. We're mammals because we have fur or hair and milk glands. We're primates, and here it says mammals with collarbones and grasping fingers. We'll go into that in a little more depth. What do we mean here? And we've already talked about that a little in class. And then we're hominids, or primates with relatively flat faces and three-dimensional vision. Within that, we have our genus Homo, which are hominids with upright posture and larger brains. We'll be getting into that in more detail in coming weeks. And then, of course, our species is Homo sapiens. So this is our position using that basic Linnaean taxonomy. And if you look at what I just described, I was describing things based on physiological characteristics. Now, Linnaeus could only base his taxonomy on physiological characteristics, like the ones described here. We call this a gradistic approach because it uses grades or levels of anatomical complexity to classify. A gradistic classification of primates might look something like this, which is a little different in the slide I gave you a couple slides ago. Here we see that primates are divided into prosimians, lower primates, versus anthropoids, upper primates. And this real divide is between like lemurs and tarsiers against monkeys, apes, and humans. This uses features to define the relationship, and one big difference is brain size in this classification. So here, brain size really pushes tarsiers to be closer to lemurs, and pushes monkeys closer to each other, and pushes humans in our own category. But there are a couple of problems with using a gradistic classification. One is this issue of convergent evolution. So what happens, for instance, if two very different species happen to evolve similar traits because they're both being selected for? What happens, for instance, if two species both tend to evolve extra fingers, and that we use that classification, the number of fingers that they have, as one way of differentiating two groups? Then we might start getting false information, false equivalencies between two very, very different species. It's also a problem of determining which traits to use. So when I gave you the violin exercise a couple of weeks ago, it was hard to tell which trait we should prioritize first. Should we group violins based off of the length of the neck, the number of strings, the number of holes? Well, we found traits that we thought these are useful traits to group and organize these violins. We weren't sure how to do it in a way that was hierarchical. Now you can imagine that this is very hard, particularly when we are still uncovering the fossil record. We're still finding evidence for some of the earliest traits. And once we find that, it can change the way that this looks. Instead, we could also use a cladistic classification, which is what we see here. And the cladistic classification uses different terminology. So instead, it uses the term strepsirrhines versus haplorhines and uses that to divide lemurs and lorises against tarsiers, New World monkeys, Old World monkeys, and ancient humans. The cladistic classification of primates is based off of clades or ancestry, using that to define relationships. Now, we're able to do that much more effectively now than we were even 20 years ago because of changes in DNA research and the number of species we've been able to track on their DNA and their genomes. But there's still some problems. First is, it reveals a lot of missing links. A lot of times, we might be able to say two species aren't quite related, but we won't know what sits between them. And it can be incredibly complex. It usually results in way more complex information because it's really tracking what you're looking at in terms of genotypes, while what we were looking at with gradistic is our phenotypes. So the cladistic classification is really looking at genetic relatedness between groups, and a lot of the variation in those genotypes will show up in a cladistic classification but be hidden in a gradistic classification. Variation within a species at the same time can be really hard to capture. So we might be able to say, hey, we've got this species, it is at the kind of top of our classification chart, and we can show how it's related to other species. But when it comes to differences in coloration, differences in traits that are being expressed that are within a species, it might not really exhibit that. And this is constantly evolving, and really this is a problem that can exist with both of them, that as we gain more evidence, we often have to reposition things in these family trees. When it comes to the hominoid classifications and what we're classifying in a gradistic versus cladistic classification system, the main difference that we can think of for us is how we posit humans against other living primates. Now, if we use that traditional classification system, we put humans in our own family that's completely separate from other living primates, completely separate from the apes. So as you can see here on the left, humans have been separated, and gorillas, chimpanzees, and orangutans are all grouped together. This is really, really different from the phylogenetic classification, which looks at DNA to see how closely related each of these groups are. And what we found from that is that humans are actually most closely related to chimpanzees and bonobos. And then this group of chimpanzees and humans is most closely then related to gorillas, and from gorillas up, we can go up a level to orangutans. So So evolutionarily, this is really helpful, but you can see there are major differences in how we're kind of organizing humans in these two systems. The other group that'll be really different in terms of how we're organizing them are tarsiers. And that's something that I want us to talk about and think about today. So today I'll describe lemurs, lorises, New World monkeys, and Old World monkeys. And at the end of our lecture, I'll give you a chance to think about how tarsiers would fit into these two family trees. And I'll tell you that in the gradistic approach, you can see on the left, tarsiers are lumped in with lemurs and lorises as prosimians. In the cladistic approach, tarsiers evolutionarily are placed closer to monkeys and much further from lemurs and lorises. So what we'll do is I'll describe the different traits that show up, and we'll use that to see what makes tarsiers more like lemurs, lorises, and what makes them more like monkeys. All right, so I've already thrown out a lot of names at you, and we're going to be doing that a lot in this lecture and some of our coming lectures. I thought it might be helpful to give you a couple just translations of many of the Latinate terms that show up when we're looking at these different groups. And the first one that's going to come up quite frequently is rhine, R-H-I-N-E, nose. So there's a lot of these that are just describing the nose shape and nose form. And once you know that means nose, it makes it a little easier to know what we're talking about. The next is anthro for man, which we already know because anthropology is the study of man. And then simian for monkey. It ends up being flat nose one point, but really monkey is commonly used for that. We also have taxonomic suffixes, ones that show up in certain parts of our taxonomy. So for instance, if you see forms at the end, it just means the shape. Oide means this resembling. Ide means kin, and ine means like. These different terms, these different endings are used to help us differentiate where something shows up in the Linnaean taxonomy. Now, I don't expect any of you to memorize all of this. I'm just giving you this as a way to help you with remembering some of the terms that show up. So we could take this same family tree, for instance, and just translate it into what we're really talking about. But for instance, primates, when we say strepsirrhenes versus haplorrhenes, which we'll talk about today, we're really just talking about nose shapes. One has a curl to its nose and the other is much simpler. Or when we look at the lemurs versus tarsiers versus monkeys, that's all we're saying is that lemuriform is a lemur form. Lemur actually means ghost, little known fact. Tarsiforms is just tarsal bone form because they have really weird tarsal bones. And simiform is just monkey form, something that resembles man if you use anthropoid, which is another term used at that level. So this is something to give you, keep it in mind. Again, I'm not going to ask you to translate all of these, but it gives you a chance to see that many of these terms that we use are not as complex as they first seem. So primates, where are primates? What does this map tell you about the evolutionary mechanisms leading to this distribution? So take a minute and look at this map and look at where primates show up along the map. Go ahead and pause the video, look at it, and just posit some hypotheses about what you noticed about their distribution. All right, so what you might have noticed is that they tend to be distributed along the equator. And they go a little bit north and a little bit south of that, but they stay primarily along the equator. So that's just something I want us to think about. We'll understand that a little bit better as the course moves on. And particularly, we'll want to think about how humans became such broad primates. I mean, we can live in any of these places now. So we'll think about how and why and when that happened within our genus, or even higher up. All right, what you might have also noticed is that we've got a clear differentiation, for instance, with New World monkeys being in the New World, apes being primarily in Africa as well as extending into, Old World monkeys being primarily in Africa and extending into parts of Asia. And then apes, if you look within that, the purple ones, you can see the orangutans are over here, but otherwise our apes are staying in Africa. So some more little hints. Now we'll start with talking about our lemurs, lorises, and tarsiers, or lemurs and lorises. And the lemurs, if you look, they only showed up on Madagascar, which is pretty cool. So there's a bunch of different species of lemurs, but they're all just here in Madagascar because we have that island effect. You remember the evolutionary mechanisms that we have can operate very differently in islands because we don't have gene flow and we have smaller populations. We also have lorises that show up both in Asia and Africa, and then we have tarsiers over here. So then let's take a chance to look at these lemurs and lorises. And let me give you a few bits of information about them, about what we use to differentiate these from other groups. And the first thing I'll point out is that lemurs today are relatively small. So if we look at their body size in comparison to other primates, they tend to be a lot smaller. Now that wasn't always true. For instance, here's an image of an ancient lemur skull. You can see it's larger than a modern day gorilla skull. So they had been extremely large in the past in some cases, but today we even have examples of a mouse lemur, which is tiny, tiny, tiny little primate. And most of the really tiny primates that we have fall into this strepsirrhine category. Now they're mostly nocturnal, and their smaller body also means they have smaller brain size. And the biggest difference, the thing that you'll notice the quickest about them, is that they have this retained snout with these inward turning nostrils, hence the name strepsis, or inward turning rhino nose. So if you look at their nostrils, you can see they're shaped kind of like a comma. And you can see they have a pretty long snout. Their snout looks more similar to that of a dog than to us. So looking, for instance, at this example and at the cranial example here, you can see the nose is quite long and extended, and it has this wet nose, like you'd see for a dog. Additionally, you might notice the eye is partially to the side rather than forward facing, which is something we see as a difference between strepsirrhines and half-marines. And there's no post-orbital closure. Instead, there's something we call a post-orbital bar. What that means is that there is a bar of bone right here that goes around the eye orbit, but it doesn't fully enclose it. So if I were to poke this individual in the eye, my fingers go right through because there's a hole in the back of their eye. Another interesting thing about them is they have an unfused mandible, so they can move their mandible while chewing. And they have this really, really cool adaptation called a tooth comb. That's what you see down here. It's always in the lower jaw. It extends, kind of juts out from underneath. All the incisors basically are really, really long. And it's a mechanism that enables them to get gums and resins from trees. They also have this really weird thing called a toilet claw. You can see it here. And they do have these kind of claw-like forms on their nails. And they're leapers. So I think the biggest one on this slide is that they're leapers. So when they move around, they can jump incredible heights. I'll show you an example of that. This is a video of a slow-moving loris. If you want to watch the video, it's quite short. Go ahead and click on the YouTube link below. Basically, I've just included it to show you how adorable lorises are. That's it. But you can see a little bit of the way that its crania is formed. You can see it has incredibly large eyes with this really dark coloration around the eyes. That's a feature that makes it very, very good at being nocturnal, very, very good at night vision. You can also get a little bit of a glimpse into its dental structure and eating habits. So go ahead and check that out if you're interested. Additionally, I have this very short link to a very homemade video of galagos, or of a bush baby, leaping around. The point of this is to demonstrate how effective these guys can be at leaping. So they can jump 7 to 8 feet from a seated position, and that makes it really easy for them to get out of the way if there's some kind of danger and to move around in the trees. The link is here. Go ahead if you want to take a peek. You can also look up videos of lemurs leaping, and get a sense of how these different species move through the trees. And lemurs are leapers as well. So we tend to see that if these individuals are moving from tree to tree, they're doing it by jumping. They're not doing it in any other way. Now, lemurs are interesting because they tend to be, not all, but they tend to be female dominant. So females in lemurs are larger than males. And they're pretty diverse morphologically because, again, we have an island effect going on in Madagascar where lemurs are. The way they help carry around their infant is by carrying them in their mouths. And so in a lot of ways, they have traits that seem a little more similar to other animals. Like, for instance, their movement, body shape can be a little closer to dogs and cats when compared to the way that other primates operate, other monkeys, or monkeys. All right. Now, strepsirrhines and haplorhines broke off around 65 million years ago. We're going to go into those timelines later. You don't have to think about that for now. And haplorhines are, in comparison, shorter nosed. So if you look at the length of the strepsirrhine nose, you can see it's quite long when compared to the haplorhine nose. It's much shorter. And we call them dry nosed. We're dry nosed because if you touch the end of our nose, right, it's dry. If you touch the end of a wet nosed animal like a dog, that's the difference that we're talking about here. So haplorhines are also referred to as dry nosed and strepsirrhines as wet nosed. If you remember, I mentioned there's a post-orbital closure in haplorhines, a post-orbital bar in strepsirrhines. That bar right here is this line of bone that goes around the outside of the eye, and there is a gap behind it in strepsirrhines. So again, I could poke my finger. I wouldn't do this, but I could poke my finger through a lemur side, get to the other side. If I were to try to do that on a person, I wouldn't get that far because we have bone that fills in that gap here. So you can see the post-orbital closure. You can see that there's bone on the back side. You can see that back here too. The plate of bone that encloses the eye, there's no plate of bone here. That's a big one because we can find it skeletally. We can find it in the fossil record. So we look for that. Another thing is that we don't have this tooth comb. The tooth comb seems to be something that was really developed within strepsirrhines, is not evidence at all in haplorhines, and may not have been around prior. That might have been something that was introduced after they divided. Haplorhines also have a single pair of nipples, whereas strepsirrhines can have multiple. That means the litter size can be larger for strepsirrhines and haplorhines. Think one pair of nipples that for each pair, the average litter size is half. So if you have a pair, you have 2 nipples, the average litter size is 1. If you have 4 nipples, the average litter size is 2. They're also larger in body and brain. So these individuals are able to have much larger brain capacity. You don't have to know this, but we think it's because the blood supply is greater, because there is a shift in how blood is being supplied to the brain that enables haplorhines to have larger brains. And they consequently also have much larger bodies on average. And haplorhines move around by brachiating or semi-brachiating. That means that they can swing in the trees using their arms or also their tails. Now within that we have the tarsiers. We're not going to talk about them yet. But we also have platyrrhines and catarrhines, which for the majority of this class I'm going to refer to as New World monkeys and Old World monkeys. Or if I write it down, I might abbreviate that to NWM versus OWM. New World monkeys versus Old World monkeys. The New World monkeys and Old World monkeys also can be differentiated based on their noses. So we use noses a lot. That's why we call them platyrrhines versus catarrhines. Platyrrhines are the ones, the New World monkeys are the ones that have their nostrils at the side. So their nose is relatively flat with the nostrils at the side. Catarrhines have a kind of curved nostril with the nose facing down. So if I had to pick a monkey's nose, this is how I would think about it. If I had to pick a monkey's nose, would I pick it from the side or would I pick it from underneath? If you pick it from the side, it's a New World monkey. If you pick it from underneath, it's an Old World monkey. If you get if you need an extra hint, just think about where do we fit in? We are catarrhines. We are closer related to Old World monkeys. And so if we had to pick human noses, you would pick from underneath, right? So our nostrils face down, we fit into catarrhines. All right. Another important difference between them is whether or not they have prehensile tails. So only New World monkeys, they're the only ones that have prehensile tails. You do not find prehensile tails outside of New World monkeys. You can find New World monkeys without prehensile tails, but if you find a prehensile tail, if they're able to grab a tree and swing with it, grab something and do something with it, any of that grabbing that happens with their tail, that has to be New World. Another thing that'll come up in this course is using dental formula to understand and differentiate species. We use dentition a lot in this class because teeth preserve very well. This is what a dentition looks like for a New World monkey. And what I want you to do is to take a second, look at the teeth, and see if you can differentiate the different tooth types. So, there are 4 tooth types we're going to talk about. There's incisors. Those are the flat teeth you have in front. They're really good at tearing things up. You have the canines. The ones that come next are sharp. They can be quite tall and long, and other primates, they aren't in humans. Then you have premolars, which you might also call your bicuspids. They're relatively symmetrical, and then you have your molars in the back, and your molars are really good at grinding things down. So go ahead and take a second, figure out your own dental formula by counting how many incisors you have, canines you have, premolars you have, and molars, taking into consideration you might be missing your third. And then count what you see on the slide here, and when you're ready, hit play. Okay, so if we look at the slide here, you can see we're going to refer to this just based off of the half, since our mouths are symmetrical. So for instance, we have 1, 2, 3, 4 incisors, but we're just going to refer to the bottom ones. So we've got 2, this is the left side, 2 incisors, 1 canine, 1, 2, 3 premolars, and 1, 2, 3 molars. Now if you check in your own teeth, depending on what's going on with your dental health, you should have a 2, 1, 2, 3 dental formula, which is just different by 1 tooth. So we have 2 incisors, 1 canine, but only 2 premolars, and then 3 molars. So it's just 1 tooth difference between New World primates and Old World primates. And it's the 2, 1, 3, 3 dental formula for New World, 2, 1, 2, 3 dental formula for Old World, and we're simply putting that formula together by counting the number of teeth for each tooth type. Okay. Now our catarrhines then are our Old World monkeys, and when I say Old World monkeys, I'm referring to this other phrase, Cercopithecus, but again, we'll just say Old World monkeys. And these are all the monkeys that we find in Africa, that we find in the Old World, that are tailed. They have to be tailed. But those tails are not necessary, they're not prehensile. So they have tails, but they cannot use them for swinging. These differentiated around 30 million years ago. Again, we'll get into those numbers in a few weeks. And they have diverse habitats. You can find them as arboreal, so you can find them just living in trees, or you can find them in a mix where they're both arboreal and terrestrial. They're able to live both on the trees and on the land. They also have really different diets potentially, so we can have some are leaf eating, others have primarily fruit in their diet. And we use some of these particularly to study behaviors of early hominids because they are along a nice point in the family tree where we'd like to see if they have certain behaviors that we can identify in the fossil record. All right, so we've made it to tarsiers after about half an hour. And what I'd like you guys to think about is what makes tarsiers more like haplorhines and what makes them more like strepsirrhines. This will give you a chance to practice what we just learned look at your notes, and then get a sense of how this cladistic-gradistic differentiation really gets messy when you actually get down and look at the details. Now, this is a tarsier. You can see it's got enormous eyes, huge legs, and its legs are particularly large because it has an unusually long talus and calcaneus. These bones we have as well. The talus and calcaneus form our ankle. And so its ankle bones are incredibly long, much longer than we find in other primates. And so that's why they're called tarsiers, because these bones are your tarsals, or the small bones that make up your foot. And it has really long tarsals, so they're tarsiers. Now, are they haploids or are they strepsirrhenes? I'll tell you, there's some traits that fall into one category and some that fall in the other. We know that they are, as I mentioned, or I did not mention, we know that they are arboreal. They are nocturnal. You can see that by their incredibly large eyes. They tend to be leapers, meaning you saw from those really big back legs, they're able to leap. That's how they move around. They have a monogamous family structure, but they have multiple in a litter. So they have multiple sets of nipples. And in terms of what they eat, they're insectivores. We can see that based off of their dentition. This is what a tarsier skull looks like. They're really small. I'll show you this skull, 3D print of this skull in the lab. It's very hard for me to 3D print even because it's so small. And this gives you an idea of what their dentition looks like. They've got these really sharp forward incisors, and then you can see the rest of their teeth, their molars, canine, etc., extremely pointed, very sharp. This makes it easier for them to eat insects. It's a better structure for eating insects. Basically, it allows them to break through all of the exoskeleton. They also have claws, as you can see here, on their hands. So, here's the question. Are tarsiers haploids or strepsirrhenes? What traits do they have? Look at what I told you. Also look for any traits that I didn't mention in this slide. Go ahead and pause the video for a second. Write down everything you can think of. Look at your notes, and then we'll discuss. Okay, so here are some of the traits that means tarsiers are more like strepsirrhenes. You saw that claw. It's very similar to that grooming claw we saw in the strepsirrhenes. And you also saw that they're really small. So when you looked at the size of the skull, hopefully you noticed the skull is actually quite small. So by body size alone, it really lets us think of them more as strepsirrhenes. They have an unfused mandible as well, and they have multiple sets of nipples. So that means that they have litter sizes that are larger. That's something that's a trait more commonly found in strepsirrhenes. At the same time, tarsiers are also more like haploids. They have a post-orbital closure. I didn't mention that, but you should have noticed it from the slides. The cranium has a plate that goes behind the eye. So if you try to poke a tarsier in its ginormous eye, you wouldn't be able to get your finger straight through because it has that post-orbital closure. They have a dry rhinarium, you might have seen in the pictures. The nose is smaller, shorter, closer to the eyes, more similar to haploids. And they don't have a dental cone, so we do not see any evidence of dental cones in their dentition. Their dentition is actually quite different from both what we've seen in strepsirrhenes and what we've seen in haploids. They're the only ones that are strictly insectivores of the groups that we've looked at. And finally, you can't see this from the slides, but the cranial blood supply comes from the promontory artery. Now that's what allows for more blood supply to the brain. We think that might account for larger brain size. It's something that's present in tarsiers, even though they still have smaller body size. So just something to think about. I hope you got some of these answers when you're thinking about the differences and similarities between tarsiers and strepsirrhenes versus haploids. In our next lecture, we're going to actually get into the apes and humans, and so get a chance to look at the primates that are most closely related to us. thanks for your time, and I will see you in lab on Friday.